Within the cellular symphony, genetic information flows seamlessly from DNA to RNA, orchestrating the intricate ballet of protein synthesis, where every codon represents a note in the melody of life.
Good day, everyone! We are the Group 4. Introducing reporters: Rose Jeline Cadalso Aceves, Gia Marie Lumaca Duran, Hannah Masamong Paranaque, Agnes Blair Quisinas, Alvimay Rivera, Dave Torres, Lucy Caliet, Jeremy Salma, Mark War, Nikki Tobio, Ivan Ilaw. You heard it right. Mr. Ilaw is one of our reporters that will give ilaw or light in our DNA to mRNA to have the protein synthesis. Now, are you all ready to hear the intricacies of protein synthesis? I guess you are. So now, you may fasten your seatbelts and open your eyes and ears as we are going to discuss the intricacies within protein synthesis. Let us now start with the first reporter. Hello everyone! Yes, it's me again. So I am Rose Jeline Aceves, the first reporter. Before I start, I will be reading for you guys the learning outcomes of this lesson. So here are the learning outcomes of our report. Lesson 8, the protein synthesis. The learning outcomes. First, illustrate the flow of genetic information. Second, distinguish between transcription and translation. Third, tell the characteristics of genetic code and list down the steps in translation and protein synthesis. Fifth, describe how recombinant DNA is formed. And sixth, explain how and why mutations occur. So now let us start with the first slide, the flow of genetic information. The central dogma of molecular biology states that genetic information contained in the DNA is transferred to RNA molecules and then expressed in the structure of synthesized proteins. So the central dogma is formulated by Francis Crick. This describes the flow of genetic information in most living organisms, essentially states that the information encoded in DNA is used to build proteins. This is the workhorses of the cell through a processes. Genes are segments of DNA that contain the information needed for the synthesis of proteins. Each protein in the body corresponds to a DNA gene. Yes, genes are the specific segments of DNA that contain the instructions for making a particular protein. Each gene encodes for a unique amino acid sequence, which ultimately determines the protein's structure and function. And yes, in most cases, each protein in the body has a corresponding gene in the DNA. However, there are some complexities in eukaryotes or the organisms with a nucleus, where alternative splicing of mRNA can lead to multiple protein products from a single gene. So, let's now move on to the next slide. Here, there are the two steps in the flow of genetic information. We have transcription and translation. First is transcription. In eukaryotes, the DNA containing the stored information is in the nucleus of the cell, and protein synthesis occurs in the cytoplasm. The information stored in the DNA must be carried out of the nucleus by mRNA. So, transcription in eukaryotes. Here, the DNA which resides within the nucleus of a eukaryotic cell serves as a template for creating a molecule of messenger RNA to mRNA. An enzyme called RNA polymerase reads the DNA sequence and builds a complementary mRNA molecule. It's important to note that unlike DNA, which is double-stranded, mRNA is single-stranded. And the next step is translation. In translation, mRNA serves as a template on which amino acids are assembled in the sequence necessary to produce the correct protein. The code carried by mRNA is translated into an amino acid sequence by tRNA. So in translation, that is also called this happens the decoding of the message. Now, mRNA enters the scene. Inside the cytoplasm, it meets the ribosome, which are the protein building factories of the cell. But mRNA doesn't directly assemble amino acids. Here, where another key player comes in, the transfer of RNA to tRNA. tRNA molecules act as interpreters, each carrying a specific amino acid attached to one end and a 3-nucleotide sequence, or the anticodon on the other end. The anticodon on the tRNA needs to match a complementary 3-nucleotide sequence, or the codon, on the mRNA for the tRNA to bind. The mRNA code is like a series of instructions, which, with each codon specifying a particular amino acid, the ribosome acts like a reading machine, moving along the mRNA one codon at a time. tRNA molecules with matching anticodons bring their specific amino acids to the ribosomes. So the ribosomes facilitates the information of a chemical bond between the amino acids, linking them into a growing polypeptide chain. This chain will eventually fold into the final functional protein. So back in the slide, the communicative relationship between mRNA nucleotides and amino acids in a protein is called the genetic code. So the genetic code is the underlying set of rules that governs this intricate dance between mRNA codons, tRNA molecules, and amino acids, ultimately resulting in protein synthesis. So, back in the slide, the communicative relationship between mRNA nucleotides and amino acids in a protein is called the genetic code. So, the genetic code is the underlying set of rules that governs this intricate dance between mRNA codons, tRNA molecules, and amino acids, ultimately resulting in protein synthesis. Before I give the floor to the next reporter, I will leave a saying that says, "In the intricate dance of life, genetic information flows like a river, shaping the course of evolution and guiding the destiny of species." Thank you. So that's it for my part. And now, let's move on to the next reporter, Miss Gia Marie Lumaca Duran. And I hope you have learned something new about my discussion.
Good ma'am. I am Gia Marie Lumacaturan. So, before I will report, I will give an inspirational message to those people who have a depression. Depression with a suffocating embrace and relentless grip and will leave you feeling powerless and adrift in the sea of despair. But I implore you to hold fast to the belief that within you resides a strength so profound, it has the power to transcend even the deepest of sorrows. Reach out to those who love you, for they are pillars of the support, steadfast in their commitment to guide you through the storm. Seek solace in the beauty of soul moments, a gentle breeze, a warm embrace, a tender smile, that remind you of the precious of life. Know that healing is not a linear journey, but a series of small steps taken with courage and determination. Embrace yourself with compassion, treating yourself with the same kindness understanding you would offer you to a cherished friend. Above all, remember that your story is not defined by your struggles, but by the resilience with which you face them. You are a warrior, a survivor, and your journey is a testament to the strength of the human spirit, with unwavering empathy and boundless compassion. So that's all, ma'am, and I will report the transcription, the RNA synthesis. Under the influence of enzyme RNA polymerase, the DNA double helix unwinds at a point near the gene that is being transcribed. The initiation sequence, only one strand of the DNA is transcribed. Peptides are linked along the DNA strand in the sequence determined by the base pairing of the DNA and the ribonucleotide bases AUGC. mRNA synthesis occurs in the 3 to 5 direction along the DNA strand, in the 5 to 3 direction along the RNA strand until until the termination sequence is reached. The newly synthesized mRNA strand moves away from the DNA, which rewinds into double helix. Synthesis of tRNA and RNA is similar to this. So, an enzyme called RNA polymerase unwinds a section of double helix near the gene that needs to be transcribed. This unwinding creates a bubble where only one strand, called the template strand, is exposed. RNA polymerase uses exposed template strand to build a complementary RNA molecule. As RNA polymerase moves along the DNA strand, it adds ribonucleotides, the building blocks of RNA. Adenine pairs with uracil and guanine pairs with cytosine. RNA polymerase switches a specific DNA sequence called the termination sequence and stop transcription. The newly formed RNA molecule called messenger RNA or mRNA detached from the DNA template. The DNA double helix forms. RNA, which is typically produced from one gene at a time. Many tRNA and RNA genes can be transcribed simultaneously. Also, tRNA and rRNA molecules undergo further processing after transcription to become functional. Transcription RNA synthesis. So this is the structure. So DNA strands unwind exposing the sequence to be transcribed. This region is labeled as the initiation sequence in the image. RNA polymerase, the enzyme responsible for building the RNA molecule, binds to the promoter sequence, which is initiation site for transcription. RNA or the messenger RNA forms based on the DNA template strand. As RNA polymerase moves along the DNA strand, it adds ribonucleotides according to complementary base pairing rules. Adenine pairs with uracil, guanine pairs with cytosine. The DNA double helix reforms. The structure incorrectly referred to the termination sequence as 3-5 and the initiation sequence as 5-3. These numbers likely refer to the nucleotide positions within the DNA strand, but they are not generally used to describe the transcription process. Introns and exons. In prokaryotic Each gene is a continuous segment along a DNA molecule. Transcription of the gene produces mRNA that is translated into protein almost immediately because there is no nuclear membrane separating the DNA from the cytoplasm. In eukaryotes, the gene segment of DNA that code for proteins, exons, are interrupted by segments that do not carry an amino acid code, introns. Both exon and intron segments are transcribed, producing heterogeneous nuclear RNA or hnRNA. A series of enzymes cut out the intron segments and splice the exon segments together to produce mRNA. Even though prokaryotic genes don't have introns, there are rare exemptions. Some prokaryotic genes, such as those in archaea, may contain introns. However, these introns are much less common than in eukaryotes. So, that's all my report, and I will give it to Hannah Paranaque.
Good morning, Ma'am. Good morning, everyone. I am Hannah M. Paranaque. Continuation of reports of Miss Gia Duran. Entrons and exons. This image depicts a conceptual diagram of the process involving entrons and exons during gene expression in eukaryotic cell. So, the top part diagram shows a segment of DNA with alternating colors to represent exon, shown in purple, and intron shown in green. Exons are segments of DNA that contain coding information for proteins, while introns are non-coding regions that do not code for amino acid. The next is the process of transcription is illustrated where the segment of DNA is transcribed into a complementary pre-messenger RNA, also known as heterogeneous nuclear RNA or hnRNA. Throughout transcription, both exons and introns are copied into hnRNA. The diagram portrays the splicing process where introns, green sections, are cut out or removed from the hnRNA. This splicing is a precise process that ensures that only the exons remain in the mRNA molecule, which is necessary for the correct synthesis of proteins. After the introns are removed, the exons or purple sections are spliced together to form the mature messenger RNA or mRNA. This splicing process brings together the exons in the correct sequence to generate a functional mRNA that carries the correct genetic information for protein synthesis. The mature mRNA then undergoes translation, a process that occurs in the ribosome of the cell. During translation, the genetic code carried by the mRNA is read in groups for 3 nucleotides or codons, and corresponding amino acids are added to the growing polypeptide chain to form a protein. So the image demonstrates the molecular process of gene expression in the eukaryotic cell, highlighting the importance of splicing to remove introns and connect exons in the correct order to produce functional mRNA for protein synthesis. This mechanism ensured the accurate. Next is the genetic code. So once the 3D structure of DNA was known, it was clear that the sequence of the bases along the backbone in some way directed the order in which amino acids were stacked to make proteins. It explained that the discovery of the 3D structure of DNA helped scientists understand that the sequence of base, the building blocks of DNA, determines the sequence in which amino acids are put together to make proteins. In 1961, Marshall Nirenberg and his co-workers began to unravel the connection between the base sequence in DNA and the amino acid sequence in proteins. It mentions the groundbreaking work of Marshall Nirenberg and his co-workers in 1961, who started a infer the connection between DNA sequence and the amino acid sequence in proteins. The genetic code uses a sequence of 3 base, a triplet code to specify each amino acid. A triplet code gives 4 cubes is equal to 64 possible combination, which is more than enough to specify the 20 amino acid. It introduced the concept of the genetic code, which uses a sequence of 3 bases known as a triplet code to specify each amino acid. There are 64 possible combinations of triplet codes because there are 4 different bases in DNA: adenine, thymine, cytosine, and guanine. The 3 positions in a codon. Each base triplet sequence that represents a codon word on mRNA molecules is called a codon. Genetic code table, which is used to translate the nucleotide sequences of messenger RNA or mRNA into the amino acid sequences of proteins during the process of translation, which occurs in the ribosome of cell. So, the left vertical axis or first letter represent the first nucleotide of the mRNA codon. The top horizontal axis or the second letter represent the second nucleotide of the mRNA codon. The right vertical axis or the third letter represent the third nucleotide of the mRNA codon. RNA nucleotides are represented by the letters U, uracil, C, cytosine, A, adenine, and J, guanine. To use the table, you would read the first base of the codon from the left column, the second base from the top row, the third base from the right column. The cell where these 3 criteria intersect gives you the corresponding amino acid that correspond to the specific codon. For example, if the codon on the mRNA is UUU, you would find U in the first column, U in the second row, and U in the third column. The intersection of these 3 positions will give you the amino acid phenylalanine. In the addition to representing the amino acid, the genetic code table also includes information about start codons, AUG, which is codons for methionine and signals the start of translation. The stop codons, UAA and UAG and UGA, which signals the end of translation. The genetic code tables help translate the language of nucleotides in mRNA to the language of amino acid in proteins, playing a crucial role in protein synthesis in living organisms. That's all. Thank you.
Hello everyone! Are you ready to continue and learn something new? I can't hear you. Okay, welcome to our video on genetic codes. Today, we'll unravel the secrets of DNA, RNA, and how they dedicate the language of life itself. Get ready to decode the mysteries of genetics and uncover the fascinating mechanisms behind inheritance. Let's dive into the world of genetic codes. The genetic code table. This table is a set of instructions that cells use to translate the information encoded in DNA into proteins. Proteins are essential for many cellular functions, and the genetic codes determines the order of amino acids in a protein. The genetic code is universal, meaning that it is the same for all living things. The table in the image shows the codons, which are the 3-letter sequences of nucleotides in DNA or RNA. Each codon codes for a specific amino acids or for a start or stop signal. There are 64 possible codons and 20 different amino acids. This means that some amino acids are coded for by more than one codon. The table also shows the anticodons, which are three-letter sequences of nucleotides or transfer RNA molecules. tRNA molecule carry amino acids to the ribosomes, where proteins are synthesized. The anticodon on the RNA molecule binds to complementary codons on mRNA, which is a copy of the DNA code. So the chart specifically RNA, which is a type of molecule that carries genetic information from DNA to ribosomes, which are the cellular machinery that synthesizes proteins. Each horizontal row represents the first letter in a codon. Each vertical column represents the second letter, and the four squares within each major box represent the third letter. The center box shows the amino acid coded for by that particular codon. For example, the codon AUG codes for the amino acids methionine, and the codon UGG codes for the amino acids tryptophan. So, characteristics of the genetic code. The genetic code applies most universally with minor exceptions. The same amino acids is represented by the same codons in all species. So the genetic codes is nearly universal. This means that with very few exceptions, the same codon will code for the same amino acids in all living organisms. This universality is a strong piece of evidence to support the theory of evolution, which suggests that all life on Earth shares a common ancestor. So next, most amino acids are represented by more than one codon, a feature known as degeneracy. So only one methionine and tryptophan are represented by a single codon. Leucine, serine, and arginine are represented by 6 codons. No codon codes for more than one amino acid. So most amino acids are encoded by more than one codon. This is known as degeneracy. For example, the amino acid leucine is coded for by 6 different codons. It is believed that redundancy helps to reduce the effects of mutation in a genetic code. If a single nucleotide is changed in a codon, it may change the amino acids that is inserted into a protein. No codon codes for more than one amino acid. It ensures that the instructions encoded in DNA are translated accurately into proteins. Only 61 of the 64 possible triplets represented amino acids, and other 3 are used as signal for chain termination. The AUG codon, or which also codes for methionine, functions as a start signal, but only when it occurs as the first codon in a sequence. So, start and stop codons. These are special codons that signal the start and stop of protein synthesis. The AUG codon codes for methionine and also acts as the start codon. 3 codons do not code for any amino acid and instead signal the termination of translation. Only 61 out of 64 possible codons code for amino acid. The remaining 3 codons are stop codons. Table 21.3. General characteristics of the genetic code. Characteristics. Codons are 3-letter words. Example, GCA is equal to alanine. The code is degenerate. Example, GCA, GCC, GCG, GCU all represents alanine. The code is precise. Example, GCC represents only alanine. Next is the chain initiation is coded. For example, AUG. Next is the chain termination is coded. Example, the UAA, UAG, and UGA. Lastly, the code is almost universal. Example, GCA is equal to alanine, perhaps in all organisms. That's all, and thank you.
Hi everyone. Hi ma'am. Good morning. Um, I am Alvima Rivera, one of the member of Group 4. So my report is translation and protein synthesis. So what comes in your mind when you heard about translation and protein synthesis? Anybody from the group? Okay. Translation and protein synthesis are fundamental processes in biochemistry that play a crucial role in production of proteins within living organisms. So there are three steps of translation and protein synthesis. So let's start. Step 1. Step 1 is the initiation of the polypeptide chain. mRNA and a small ribosomal subunit join the initiating codon, or AUG, is aligned with P peptidyl site of subunit. Uh, TRA, tRNA brings a methionine karyothics or N-formal methionine prokaryothics. The resulting complex binds the large of ribosomal subunit to form a unit called the initiation complex. So, namin makita diha nga kanang sa ubos sa iyang nga figure is na diha ang ribosome nang ay blue o red. Ang blue is a small subunit which ang red kay large subunit. So these steps involves the coming together of several components necessary for the translation process begin. Components involve the initiation's mRNA, a molecule carries the genetic information copied from the DNA in the form of a series of codons, each of which specifies a particular amino acids. Kanang a small ribosomal subunit is a part of the ribosome, the cellular machinery that synthesizes proteins. It binds to the mRNA initiating codon. This codon is initiator tRNA transfer RNA molecule carrying the amino acid methionine. It binds to the start codon and the mRNA to initiate translation. At the formation of initiations complex, the small ribosomal subunit mRNA initiator tRNA and other initiations factors came together to form the initiation complex, which marks the beginning of translation. The result of this, the initiation complex is ready for the elongation phase of translation, where ribosomes will move along the mRNA, reading the codons and bringing in tRNA molecules carrying the corresponding amino acids to build a polypeptide chain. Okay, so let's proceed to step 2, elongation of the chain. The next incoming tRNA with the anticodon that is complementary to the mRNA codon bonds at the amino acyl site on the mRNA. A peptide bind is formed between the amino acid segments catalyzed by peptidyl transferase, which release the amino acid chain from the P sites. The elongation of the polypeptide chain. This step follows the initiation of the protein synthesis process. Once the ribosomes has been assembled with the mRNA and the initial tRNA, the elongation process begins. tRNA and codon binding a tRNA carrying an amino acid. In this case, phenylalanine, abbreviated as a Phe, enters a site of the ribosomes, the tRNA has an anticodon that is complementary to the mRNA codon. For example, the aminoacyl tRNA for phenylalanine has the anticodon UUC, which pairs with the codon AAG on the mRNA. Peptide bond formations. After the correct tRNA pairs with the mRNA codon is the A site, a peptide bond forms between the amino acid on the A site tRNA and the P site of the ribosomes. The translocation. Once the peptide bond is formed, the ribosome moves along the mRNA strand in a process called translocation. The tRNA that was in the P site now, without its amino acid, moves to the E site and then exits the ribosome. The tRNA and the A site now moves to the P sites, carrying the growing polypeptide chain with it. So, balik-balik ko ang process. This entire process of tRNA binding, peptide bond formation, and translocation is repeated for each new amino acid added to the growing polypeptide chain, elongating it until a stop codon is reached on the mRNA. So, this is the continuation of elongation of the chain. The empty tRNA release and the whole ribosome moves on the codon along the mRNA towards the 3-end translocation. Another tRNA attached to the A site, and the elongation process is repeated. So, translocation is the process by which messenger RNA or mRNA is decoded by a ribosome to produce a specific amino acid chain or polypeptide that will later fold into an active protein. Elongation of the polypeptide chain is continued in this step. The process starts with an mRNA sequence and a ribosome with two sites, P and A sites, for transfer RNA, tRNA molecules to bind. On the left side, the first tRNA is the P site with the amino acid methionine meet, while the second tRNA with the amino acid phenylalanine Phe is in the A site. Codons on the mRNA specify which amino acids are added to the chain, with AUG coding for meat and AAA coding for Phe. A peptide bond forms between the methionine and phenylalanine amino acid, connecting them together. The ribosome moves along the mRNA in the 5 to 3 direction from the A site to the P site as a new amino acid is added to the growing polypeptide chain. As the ribosome advances, the tRNA in the P sites moves to the E site and is released, while the tRNA in the A site moves to the P site to make room for the next tRNA carrying the next amino acids. The process repeats with each new codon on the mRNA being translated into its corresponding amino acid until a stop codon is reached, signaling the end of translocation and the release of the complete completed polypeptide chain. This figure illustrates the process of translocation during protein synthesis, specifically showing the elongation step where amino acids are added to the growing polypeptide chain. Understanding this fundamental process is crucial for the grasping and the complexity of the gene expression and protein production within cells. So the remaining step will be discussed to Mr. Dave Torres. Thank you.
Good morning, everyone. So my name is Dave Torres. So this is the continuation of the report of Miss Albe Rivera. So my topic is the step 3 of the termination polypeptide synthesis. Termination of polypeptide synthesis. Steps 3. The elongation continues until the ribosome complex reaches a stop codon. This step occurs when the ribosome complex reaches a stop codon in the mRNA sequence. Termination factors proteins bind to the stop codons and separates the protein from the final tRNA. Once the ribosomes reaches the stop codons, a termination factor protein binds to the stop codons. This binding causes the ribosomes to separate the newly synthesized protein from the final transfer RNA-tRNA molecule. After the protein is released, the ribosomes is free to synthesize another protein molecules. The ribosomes can dissociate into its subunits and be reused in the process of translation. During translation, several ribosomes can move along a single strand of mRNA simultaneously, producing multiple identical proteins. This simultaneous translation by multiple ribosomes is known as polyribosomes or polysomes. As the polypeptide chains emerges from the ribosomes, it starts to fold into its secondary structure, which refers to the folding of the chain into alpha helices, beta sheets, and other structural elements. This folding is driven by the interaction between the amino acids residues in the polypeptide chain. Finally, the polypeptide chain continues to fold and form the complete protein, adapting its characteristic three-dimensional shapes. The folding process is spontaneous and results in the protein functional structure. During the termination steps of protein synthesis, the ribosomes reaches a stop codons. A termination factor protein binds to the stop codon, and the protein is separated from the final tRNA. The ribosomes can be synthesized another protein molecule, and the growing of polypeptide chain spontaneously folds into the characteristic 3D shape of the protein.
Good morning, ma'am. My name is Jose Calyet Jr. So for today's video, I will discuss my report about the mutation. So mutation are any change resulting in an incorrect base sequence on DNA. So mutation mao ang bisan unsa nga kanang kausaban nga moresulta sa usa ka sayop nga base sequence sa DNA. Then even though the base pairing mechanism provide nearly perfect way of copying DNA, on average 1 of every 10 bases are copied incorrectly. So bisan pag ang mechanism sa pagpares sa base naghatag og kanang hingpit nga paagi sa pagkopya sa DNA sa average usa sa matag 10 base sayop nga gikopya. Then this leads to a change in amino acid sequence in protein or causes the protein not to be made at all. So kanang nagdala sa pagbag-o sa kanang sunod-sunod sa mga amino acid sa usa ka protein, o hinungdan nga ang protein dili gyud kanang mahimo. Mutation occur naturally during replication. They can also be included by environmental factors, ionizing radiation, X-ray, UV gamma rays, mutagens which are chemical agents. So ang mga kanang mutation natural nga mahitabo sa panahon sa kanang pagkopya. Mahimo na siya usab kini sa kanang sa mga hinungdan sa kanang factors. Then mutation may be beneficial to an organism by making it more capable or surviving in its environment. Ultimately, over millions of years of accumulating chains leading to the evolution of new species. So ang mga mutation mahimong kanang mapuslanon sa usa ka organism pinaagi sa paghimo sa nga mas makahimo nga mabuhi pa sa iyang palibot. Then sa kapin sa kanang milyon katuig nga na kanang naipon nga pagbag-o nga nagdala sa kanang evolution sa bag-ong mga species. Then since much of an organism DNA does not code for anything, mutation in this region are neutral. Other mutation can be harmful, either producing genetic diseases or other debilitating condition. So kanang tungod kay kadaghanan sa DNA na sa usa ka organism wala mag-code bisan unsang kanang bisag unsa nga butang ang mga mutation. So kini nga mga region neutral. Ang ubang mga mutation mahimong makadaot, mahimong makapatunghag genetic nga mga sakit o uban pang makapaluya nga mga kahimtang. And that's all. Thank you, ma'am.
Good day, everyone. So I will be reporting recombinant DNA up to the restrictions of enzymes. Recombinant DNA is created by inserting DNA segments from one organism into another's genetic materials. In this case, genetic engineering of E. coli involves adding the gene for human insulin production, allowing for the mass production of insulin to treat diabetes. So, in Table 21.4, this table lists substances produced through genetic engineering and their uses. So, human insulin treats diabetes mellitus. Human growth hormone treats dwarfism. Interferon fights viral infections. Hepatitis B vaccine provides protection against hepatitis B. Malaria vaccine provides protection against malaria. And tissue plasminogen activator promotes the dissolving of blood clots. So as you can see in the slides, it explains how restriction enzymes, primarily found in bacterial cells, function to cleave DNA molecules, except for those with specific modifications like methyl groups. These enzymes help protect bacteria from foreign DNA invasion by cleaving DNA lacking these protective modifications. Here it describes how restriction enzymes work on DNA. They recognize specific sequences called palindromes, where the DNA sequences reads the same forward and backward on complementary strands. They cut the DNA at these points, creating fragments with known sequences that can be manipulated using DNA ligases. So that concludes my report. Thank you everyone and God bless.
So good day everyone, I am Mark J. Guerso, member of Group 4. So today, I will discuss my topic, the plasmids and where plasmids is isolated. So let's start from the plasmids. So plasmid, the introduction of this is the new introduction of a new DNA segments called the gene is into a bacterial cells require a DNA carrier called a vector, which is often a circular pieces of double-stranded DNA called a plasmid. Plasmids range from 2,000 to several hundred thousands of nucleotides and are found in the cytoplasms of bacteria cells. Plasmids function as the accessories to chromosomes by carrying genes for the inactivation of antibiotics and the production of the toxins. They are also able to replicate independently of chromosal DNA. So, therefore, plasmids are circular pieces of double-stranded DNA that serve as a carriers of or vectors for introducing new DNA segments such as genes into a bacterial cells. They are found in a cytoplasm bacterial cells and range in size of 2,000 several hundreds of nucleotides. Plasmids play a role as accessories to chromosomes by carrying genes that and can inactivate antibiotics or produce toxins. Additionally, plasmids have ability to replicate independently of the chromosal DNA. Plasmids can carry also the antibiotic resistant genes, toxin genes, metabolic genes, virulence genes, conjugation genes. It is important because the specific genes carried by plasmids can vary greatly depending on the bacterial species and the environmental condition. So let's tackle about the formation of recombinant DNA. So it is a plasmid isolated from a bacterium and a restriction enzyme is added which leaves it at a specific site. The GGATCC a restriction enzyme. Then GATCC and CCTAGG. CCTAG are the sticky ends. When the circular DNA is cut into two sticky ends are produced which unpaired bases which produce unpaired bases. The sticky ends are provided with a complementary section for pairing from human chromosomes to which the same restriction enzyme has been used. The GATCCG. Human DNA segment is the GCCTAG. Okay. So the explanation of this is the isolation of a plasmid is a form from a bacterium. Plasmids are circular pieces of DNA found in a bacterial cells. Cleavage with restriction enzymes is added to the plasmids. Restriction enzymes are the proteins that can recognize as a specific DNA sequence and cut the DNA at those sites. In this case, the restriction enzymes cleaves the plasmids at a specific sites resulting into two fragments. The sticky ends, the cleavage of plasmid produces two fragments with unpaired bases. These unpaired bases are called the sticky ends because they can form hydrogen bonds with the complementary sequences. The complementary section of a human DNA segment is used which has been cut with the same restriction enzyme. These human DNA segments also are sticky ends with the complementary sequences to the sticky ends of the plasmids. Pairing of sticky ends, the sticky ends of the plasmids and the human DNA segments can pair together through complementary base pairing. In the provided explanation, the sticky ends of the plasmids is the GATCC pair with the complementary sticky ends of the human DNA segments of GCCTAG. Formation of the recombinant DNA is a pairing of sticky ends of the plasmids and the human DNA segments result in a formation of the recombinant DNA. The plasmids now contains a section of human DNA creating a hybrid molecule.
Good day, everyone. So, my topic is all about the formation of recombinant DNA. So, the formation of recombinant DNA. A plasmid is isolated from bacterium and a restriction enzyme is added which cleaves to a specific site. So, kuan, mo na siya i-picture sa katong restriction kanang specific site nga siya GGATCC. Then mo agi siya restriction enzyme, tapos mo na siya resulta nga G sa babaw CCTAG. Then naa siya sticky ends sa ilalom sa iyaha, murag siya line. Tapos GATCC G sa lalom. So tapos na siya sticky ends gihapon sa ng iyang ilalom. So with the circular DNA is cut, 2 sticky ends are produced. So mo to siya. Duha na ka sticky ends ang kuan katong sa una og ikaduha kay kuan man siya gi-cut man siya into 2. Then which have unpaired bases. So unpaired. Thus sticky ends are provided with complementary section for pairing from a human chromosomes to which the same restriction enzyme has been used. So, kato ang sticky ends is provided na siya with complementary section. Kay after that nga gi-cut siya, kuan man siya, i-pair man siya sa human chromosomes. Nga then ang gi-cut sa human chromosomes o ang katong plasmid is ang restriction enzyme. So, mo na siya picture. Mo na siya GATCC. So, human sa GATCC sa G sa ilalom, tapos may human DNA segment. Tapos G sa taas was CCTAG. Then the breaks in the strand are joined using DNA ligase. So, gi-break sila using the DNA ligase. And the plasmid becomes a circular piece of double-stranded recombinant DNA. So, mao to siya. Ang katong gikuha nga part sa plasmid o sa kanang human chromosome is gi pair siya kanang ganing gi-join, giusa ba siya? Giusa, tapos mahimo siya nga recombinant DNA. So, mo na siya kuan, bacteria. Tapos aiyahang mo na siya picture sa bacteria. Tapos sa sulod niya is ang plasmid. So if ang usa na nga circle nga usa ka plasmid is isolate kuan siya ni murag gikuha siya is mo na siya gitawag nga isolated plasmid. So mo na siya picture isolated plasmid. So the plasmid is cleaved with the restriction enzyme. So mo siya gikuha by restriction enzyme. Then ang kuan ato siya, ang gikuha nga part ato siya, maputol ang iyang circle. So ang kaputol ato siya nga circle is naa siya sticky ends. So duha ang iyang sticky ends. Pareha sa ato ang kuan ganiha katong naay gipakita nako nga picture. Tapos human chromosome. So human chromosome mo na siya kaning blue. Mo na siya picture. Human chromosome desired gene is clipped out with the same restriction enzyme. So magkuha gyapon og kanang part sa human chromosome para mapares sa ato ang plasmid. Pero ang gamit gyapon nila is restriction enzyme. Desired gene and plasmid are spliced together by DNA ligase. So giusa na ang plasmid og ang human chromosome gamit ang DNA ligase. So ang resulta is recombinant DNA. So mao na siya recombinant DNA, imbis ang circle ta ganiha sa plasmid ra unta to is perfect circle yellow or orange ba. Tapos cut man siya, then giadan og human chromosome, so mo na siya naa siya blue. So mo na siya gitawag og recombinant DNA. Bacterium with recombinant DNA molecule. So mo na siya kanang picture. Makita nimo siya nga na siya tulo ka circle tapos na siya tulo ka plasmid nga color yellow or orange, then naa siya usa ka circle nga kuan na siya half nga orange or yellow tapos may blue. Mo na siya kanang may blue ana is mo na siya gitawag nga recombinant DNA. So mo na siya bacterium with recombinant DNA. So the bacteria reproduce and they replicate all of the genes including the new recombinant DNA. So ang bacteria di is mo reproduce pud sila, mo replicate pagyud. Then ilang i-copy tanan kahasta tong naa na siya recombinant DNA. Because bacteria multiply quickly, so ang bacteria dali kay siyang kuan kanang multiply din siya. Every second maka-multiply siya. There are a soon large number of bacteria containing the modified plasmid. So mo na to siya. Ang modified plasmid is mao tong recombinant DNA. Then mo kon siya daghan na kayo siya nga modified na nga kuan plasmid. So which are capable of manufacturing the desired protein. So sila ang katulad siyang recombinant DNA is capable siya for manufacturing the desired protein for the body. So maora toy akong part.
So good morning everyone. My name is Ivan Ilaw and I will continue the report for the formation of recombinant DNA. So when the bacteria reproduce, they go through the process called replication, where they duplicate all of their genes, including the newly introduced recombinant DNA plasmid. These plasmid are small, circular pieces of DNA that carry specific genes. Bacteria have the ability to multiply rapidly through the process called binary fission. During binary fission, a bacteria cells divide into two identical daughter cells, which daughter cells receive a copy of genetic materials, including the replicated genes and plasmid. Due to the fast multiplication rate, bacteria quickly produce a large number of offspring. As a result, there is soon a large population of bacteria that contain the modified plasmid. This means that a significant number of bacteria are capable of manufacturing the desired protein coded by genes on plasmid. So next is the recombinant DNA technology. So recombinant DNA technology is a technology mainly used to change the phenotype of an organism or host when genetically altered vector is introduced and integrated into a genome. So basically this process involves the introduction of foreign pieces of DNA structure into the genome which contains a gene of interest. This gene which is introduced is the recombinant gene, and the technique is called the recombinant DNA technology. There are multiple steps or tools and other specific procedure followed in recombinant DNA technology, which is used for producing artificial DNA to generate a desired product. So these are the steps for the recombinant DNA technology. So first step is the isolation of genetic materials. So the first and the initial step in recombinant DNA technology is to isolate the desired DNA in its pure form, free from other macromolecules. So step 2 is cutting the gene at the recognition site. The restriction enzyme play a major role in determining the location at which the desired gene is inserted into vector genomes. These reactions are called restriction enzyme. So step 3 is the amplifying the gene copies through polymerase chain reaction, or known as PCR. This is a process to amplify a single copy of DNA into thousands to millions of copies once the proper gene is interest has been cut using restriction enzyme. So next is step 4, the ligation of DNA molecules. In this step of ligation, the joining of two pieces, a cut fragment of DNA and the vector together with the help of enzyme DNA ligase. So last is the step 5, insertion of recombinant DNA into host. So in this step, the recombinant DNA is introduced into a recipient host cell. This process is termed as transformation. Once the recombinant DNA is inserted into the host cell, it gets multiplied and is impressed in the form of manufactured protein.